Everybody, my name is Asha and I'm from the University of Glasgow in Scotland. So I'm just going to give you a quick introduction about my PhD project and hopefully you can have a look at my poster then. So I am doing a microbiome project. As you know, microbiome projects have exploded in the last 5-10 years, so it is a very hot topic. My title is Invasions of the Pharynx, Microbiome of Infected Respiratory Tissue. And I am interested in the ecology of microbial communities, looking at stability, diversity, and variation in bacteria from the pharynx in healthy and disease-related subjects. So initially, we took pharynx swabs from healthy participants. Okay, we can go into the next slide, that's fine. So we took pharynx swabs from healthy participants over a defined period of time. When the participants got sick, for example, with a common cold or infection, we identified these as unhealthy samples. We sequenced the V1, V2 region of the 16S gene through MySeq sequencing. So what we found was that overall, each individual had a unique microbial community structure. But looking at the results here, the majority, well actually all of the healthy samples were firmicutes dominant, whereas the unhealthy samples had cases of protobacteria dominance, so there was a shift. So the firmicutes are in the purple and the protobacteria is in the brown colour. We also found that participants were very quick to recover from a cold or infection, so that the community structure after infection was similar to what it was before the infection, so they were highly resilient. This could be because of the age of the participants, but also the overall health status. I'll just finish up there. I mean, my poster is there for you to have a look at, and feel free to ask any questions if needed. So, thank you.
Okay. Next up.
Okay, so thank you for this opportunity. My colleagues and I at University of California, Irvine completed the experiment Age-Dependent Patterns of Adaptation in Drosophila, the common fruit fly. So as we're all aware, there's the paleo hypothesis of health that claims that, of course, we should revert to a Paleolithic diet to see, health advancement or to alleviate many chronic disorders. Against them, there are those, like was discussed in Marlene Zook's talk, that long agricultural populations are well adapted to the agricultural diet due to the speed with which natural selection can foster effective adaptation to novel environments or diets. So I would like to bring your attention to a third hypothesis that's based on William Hamilton's 1966 Forces of Natural Selection, which kind of digresses from both of these views. Okay, it states, or essentially, populations can adapt quickly at early ages to a novel environment, but only slowly or incompletely at later ages. So numerical calculations support this general early weighted adaptation hypothesis. So what we did is we completed diet manipulation studies on very large cohorts, highly replicated, on the fruit fly, in which we actually exposed them to an evolutionarily recent diet, an imposed diet through domestication to our lab, as well as an ancestral diet. And the results seem to conform or provide evidence for the Hamiltonian hypothesis, with populations actually performing better on their ancestral diet, however, only at later ages. Okay? So this could possibly suggest or provide a suggestion that humans can actually revert to a Paleolithic diet at later ages, but at early ages we might see actually good health on an agricultural diet. Thank you.
Thank you. Next up, harnessing whole genome sequence data to detect mixed infections in ticks.
Hi, I'm Catherine Walter, and I'm a member of the Duke Wasser Laboratory. Hosts, whether they are humans, other vertebrates, or arthropod disease vectors, are infected with often large, often heterogeneous populations of pathogens. Ticks are often co-infected with multiple pathogen species in addition to multiple strains of the same bacteria. However, the role of mixed strain infections of Borrelia burgdorferi, the bacterial agent of human Lyme disease, in shaping human disease outcomes and in driving pathogen evolution is unknown. Bacterial genotyping via Sanger sequencing is reductive, and traditionally, single ticks have been genotyped with a single infecting Borrelia strain. However, deep population sequencing allows us to generate a portrait of the Borrelia burgdorferi diversity present within individual field-collected tick vectors. Here we take a deep sequencing approach, and we find a high prevalence of mixed Borrelia strain infections. We also find a signature that within vector diversity is composed of both diversity present in a complex inoculum, that is the blood meal which the tick is infected by, as well as continued diversification within the tick. So population approaches will allow us to explore potential facilitation or competition within individual ticks or hosts, as well as explore the role of multiple strain infections in shaping clinical outcomes.
Great. Next up, parent of origin effects in Turner syndrome patients.
Hi, my name is Jada. I'm currently a freshman undergrad student at Arizona State University. And so generally, in a general sense, Turner syndrome patients have one X chromosome without a partner X or Y. And it has been suggested that based on the inheritance of maternal X chromosome or paternal X chromosome, it affects the severity of the Turner syndrome as well as the incidence of mental disorders. So in this research, we conducted analysis on the current research on Turner syndrome, trying to determine the effect of paternal X versus maternal X and its effect for physical traits as well as mental disorders or psychiatric traits, and with the goal to clarify the parent of origin effects in those patients. And a thing to know is that the alleles with parental bias may also affect the development. So overall, the evidence for parent of origin effect isn't as strong as we had hoped. And but in a general sense, the paternal X chromosome showed a more of a physical trait differences, as maternal X chromosomes show more of a cognitive differences. Thank you.
And last but not least, evolving microbiota and the coevolution of sex and aging.
Hello, I'm Milind from India. I believe that evolutionary medicine is not only application of evolution to medicine, there needs to be a reverse flow as well. So thinking about disease can help us in refining, revising, and at times even radically altering evolutionary theory. And here is one example where we play with the idea how infections interact with the aging process. It stems from Hamilton's theory that the microbiota, the parasites on our body, are evolving within a host lifetime. And the counterpart of this remains unexplored. That is, how the evolving microbiota crosstalks with the aging process of the host body. And we model this situation using substantial mathematics, which is there in the poster. And I will just mention two results. One is the interplay of evolving microbiota and the host results into a negative frequency dependent selection where genotypes with different lifespan can coexist. And number two, it gives rise to an optimum lifespan, and this lifespan is decided by two parameters. One is if there is a kin bias in transmission, and the other is the death-related parameters or the fitness disadvantages of the parasite. So you're all welcome for the poster. Thank you.